Hi everybody, this is Miss Smith and this is the flip video for cells part 1. Today we're going to be looking at some of the 6th grade things that you should know from 6th grade that are going to help you to be able to do some of the 7th grade things about cells that we're going to learn this year. Well, let's start out first of all with a very basic definition of a cell. A cell is the basic unit of structure and function in all living organisms, and this right here is a picture of a cell. Okay, so today what I want you to do is you're going to take some Cornell notes in your ISN. Make sure you have talked with your teacher so you know what page to be on. And we're going to go ahead and do a little review of what Cornell notes should look like to start out with. So this is going to be just a quick reminder about what Cornell notes should look like. Remember, Cornell notes should be on the right side of the page, as this is something that is from your teacher. So just as a quick reminder, the left column here and the summary down here at the bottom should be done after your notes. The bulk of your notes should be taking place over here in the right column. So make sure that you're writing like you text. Do not write every single thing that the teacher says. Make sure that you're writing the big ideas. If you overwrite, if you go back and make sure that you highlight things that are important or underline the most important things in your notes. But keep in mind to write like you text. Summarize, abbreviate as much as you can. So after you've taken your Cornell notes, and I encourage you to do it in kind of an outline form. So let me just kind of minimize this here. I encourage you to kind of do it in an outline form. For example, if I'm writing here, I'm writing along, and then I have a key point, maybe put a bullet point and then make some more text. If there's information that's underneath that, you can kind of have sub-bullets underneath. And then if you go to the next big idea and write it above and then have some sub-points underneath that, that's how I would encourage you to take your notes. This is probably the best way to do so. Okay, so if I'm going to come back here to my Cornell notes, about 24 hours after you have watched this video, I want you to go back and I want you to work on your left column and your summary column. So your summary, remember, should be short and sweet. It should summarize what you learned, kind of the big points from the notes, so that when you go back to study, you can kind of read your summary to see what is in the notes above it. Then in your left column here, this is for key vocabulary and for House of Rigor questions. So let's review what a House of Rigor question looks like. On page 4 of your ISN is where your rigor question words are. So remember, when you're writing a House of Rigor question, you want to kind of stick with the basic questions first, because the basic questions are much easier to write. As you improve on your House of Rigor questions, you can move up to level 2 and even level 3. For these notes, I would like you to write 2 level 1s. So I want you to write two level 1s here, and I want you to write 2 level 2s. Okay, so in your left column of these notes is what you're going to be writing those on. Remember that a House of Rigor question is something that your teacher may ask you in class. So make sure that when you're writing your House of Rigor questions, that you're thinking of what are things that I want to know from these notes that are going to help me study. Okay, so make sure you're taking your Cornell notes throughout. Let's go ahead and get started talking about cells. Okay, so let's look at what we're going to cover today. So your objective should be written at the top of your Cornell notes, and this today is our student expectation. So let's look at what our objective is. Our objective is that the student is expected to understand that all organisms are composed of one or more cells, and recognize that the presence of a nucleus determines whether a cell is prokaryotic or eukaryotic. So already we have a few keywords you could put in your left column. Those include cell, prokaryotic, eukaryotic, and nucleus. Now let's look at our key concepts for this video. Each of these key concepts could be a bullet point in your Cornell notes, so you could have 6 major bullet points with some sub-notes under each one. Okay, so let's look at what those key concepts are. Number 1, the cell is the basic unit of life. Key concept 2, all living organisms are composed of one or more cells. Key concept 3, eukaryotic cells contain an organelle called a nucleus and other membrane-bound enclosed organelles. Prokaryotic cells do not contain a nucleus. Key concept 4. The membrane-enclosed nucleus in eukaryotic cells contains DNA, or the genetic information within the chromosomes. The DNA of prokaryotic cells does not form a chromosome, nor is it contained within a nucleus. Key concept 5. Taxonomic classification is a process of science that organizes living organisms by their structure, function, and relationships. And finally, key concept number 6, autotrophic organisms manufacture their own nutrients, where heterotrophic organisms must consume other organisms to obtain life-sustaining organic molecules. So make sure in your Cornell notes that you are writing down these key concepts, and make sure that you're using your left column for vocabulary words, and at the end, House of Rigor questions. Okay, so we're going to start with prokaryotic cells. Cells that lack a membrane-bound nucleus are called prokaryotics. It's from the Greek word meaning nuclei. These cells have very few internal structures that are distinguishable under a microscope. You can't really see them. Cells in the Monera kingdom, such as bacteria and cyanobacteria, also known as the blue-green algae, are prokaryotic cells. They differ very significantly from eukaryotic cells because of a couple of important features. Number 1, they are unicellular, which means they're single-celled organisms. They are found in all environments, whether it be land or water or the different types of biomes within the land and water, so deserts and rainforests, they're found all over the place. They're also the largest group of organisms, mostly because most of them are bacteria. Because bacteria is so small, there's high amounts of bacteria. Also, the biggest characteristic of a prokaryotic cell is that it lacks a nucleus, which means there's no nuclear bound membrane inside of the cell. The genetic material is just all over the place. In other words, they also do not make any kind of chromosomes with that DNA. So it's different than the types of cells that you and I have. There is also no membrane bound organelles also, which also makes a big difference. There are 3 types of shapes that a prokaryotic cell can be found in: rod-shaped, spherical, and spiral. So these are the shapes of our prokaryotic cells here. Let's go ahead and take a look at these. There's 3 shapes: spherical, and those spherical ones are here. Rod-shaped, a rod means it's long. And spiral could be like this. It could also be around in a circle. So those are the 3 types of shapes that prokaryotic cells can come in. Okay, the next kind of cell we're going to look at is a eukaryotic cell, or eukaryotes. Eukaryotic cells come from the Greek meaning truly nuclear, comprise all of the life kingdoms except Monera. In other words, that's the bacteria. They can be easily distinguished through a membrane-bound nucleus. So eukaryotic cells also contain many internal bound structures called organelles. These organelles, such as the mitochondria or the chloroplast, serve to perform metabolic functions and energy conversion. Other organelles like the filaments and the ribosomes have other functions that we're going to talk about in one of the later videos coming up. So again, they can either be multicellular or unicellular. They are all of the plant and animal cells that we will talk about. They have a nucleus. In other words, they have a nuclear membrane that surrounds the DNA. So if I can make this a little bit bigger, you can see here that this is what I'm talking about when I say a nucleus. And inside the nucleus is where we see our DNA, or our genetic information. This here would be a animal cell. Okay, and then our other kind is a plant cell. And again, the plant cell, we see there's a membrane-bound nucleus, and inside of it we have the DNA. Okay, so that is our type of eukaryotic cells. So as you can see in those pictures, they have a lot of organelles, and you can see it's very complex. If you go back and look at prokaryotics, they look very simple. Also, under a microscope, you can view the different organelles. So you can see under a microscope, under very high power, these different organelles that we're talking about. Organisms are divided into autotrophs and heterotrophs according to their energy pathways. An autotroph, in other words, plants, algae, and many bacteria, are self-feeders, also called producers. They produce complex organic compounds such as carbohydrates, lipids or fats, and proteins. These autotrophs use simple substances present in their surroundings. Generally, they get their energy from light, in other words, they're phototropic, or they get their energy from inorganic chemical reactions. In other words, they go through chemosynthesis. So again, these are the producers in our food chains, like the plants and algae. And again, they're able to make their own food. They don't need living energy or an organic carbon source. They can make it themselves. Autotrophs reduce the carbon dioxide that we see in that we get from autotrophs in order to make those organic compounds. Heterotrophs, or another word for that would be a nutrition, is an organism that cannot fix carbon or use organic carbon for growth. This is different than an autotroph because an autotroph can use those different energy sources to make organic compounds. A heterotroph cannot do that. A heterotroph has to use those organic compounds in order to breathe and have different life processes. So animals, fungi, and other many different types of bacteria use those organic compounds that autotrophs make in order to have energy. They then release carbon dioxide back into the environment, in which the autotrophs then will use in order to continue the cycle. Scientists have found and described approximately 1.75 million species on Earth. That's a lot of species. Plus, there's new species being discovered every day. From tiny bacteria to yeast to starfish to blue whales, life's diversity is really impressive. So with all this diversity on Earth, how do we make sense of it? Well, one way to make sense of all of these different creatures is by classification. Scientists put similar species into groups so that these millions of species do not seem to be so overwhelming. Some people rely on their knowledge of classification to understand what different species are. For example, you may have done this without thinking about it. Let's say your friend comes to you and says he saw an egret last weekend in the bayou. You may have never heard of an egret, but if he tells you it's a bird, you may have some idea on what it looks like. In fact, this is an egret. So if you know that that's a bird, that would help you better to understand and classify it. Living things are divided into three groups based on their genetic similarity. The three groups are Archaea, Eubacteria, and Eukaryota. Okay, our Archaebacteria are very ancient prokaryotic microbes. Eubacteria are more advanced prokaryotic microbes, and Eukaryota are all life forms with eukaryotic cells that include plants and animals. So these are all called domains. The figure at the left shows the three domains of life. The distance between the groups indicates how closely related they are. Groups that are close together, like plants and animals, over here, like plants and animals that are grouped close together, are much more closely related than groups that are further apart, like plants and the bacteria that are down here. We're going to look at the Eukaryota domain because it's divided into several groups called kingdoms. When the first system was developed, there were only two kingdoms, plants and animals. But with the discovery of the microscope and leading to the discovery of more organisms, there was a new system that needed to be put in place. So now there are 6 kingdoms. So the first kingdom we'll look at will be plants. Plants are autotrophs, which means they can make their own food. Plants are all multicellular and consist of complex cells. There are over 250,000 species in the plant kingdom. It is the second largest kingdom. Plants range from tiny little mosses to giant trees. But remember, without plants, life on Earth wouldn't exist. Plants feed almost all of the heterotrophs, organisms that eat other organisms on Earth. The animal kingdom is the largest kingdom with over 1 million known species. All animals consist of many complex cells, and they are heterotrophs. Members of the animal kingdom are found in the most diverse environments of the world. Archaebacteria are the third type of kingdom. In 1983, scientists used samples from a spot deep in the Pacific Ocean where hot gases and molten rock boiled into the ocean to form the Earth's interior. To their surprise, they discovered unicellular or single-celled organisms in those samples. So those are classified as archaebacteria. They are found in very extreme environments such as hot boiling water and thermal vents under conditions with no oxygen or highly acidic environments. Eubacteria are like archaebacteria in that they're complex and single-celled, but most bacteria are in the eubacteria kingdom. There are kinds found everywhere, and the only ones that people are familiar with are very small. Eukaryotic are classified in their own kingdom because their chemical makeup is different. Some types of eubacteria are vitamins and foods like yogurts, but some are also bad for you that can give you strep throat. Our next category is going to be fungi, which are made up of mostly mushrooms, mold, and mildew. Those are all examples of organisms in the kingdom fungi. Most fungi are multicellular and consist of complex cells. Fungi are organisms that biologists once confused with plants. However, unlike plants, fungi cannot make their own food. Most obtain their food from parts of plants that are decaying in the soil. So most of them are decomposers. Our final and last kingdom are the protists. Slime molds and algae are protists. Sometimes they're called the odds and ends of the kingdom because its members are so very different. Protists include all microscopic organisms that are not bacteria, not animals, not plants, and not fungi. So they're kind of the last little bits. Most protists are unicellular, which means they're single-celled, and they are not classified into the bacteria kingdoms because, unlike bacteria, protists are complex cells. So an example of that would be diatoms that are found in the ocean. The last portion of this video is going to talk about something called cell theory. In 1665, when the microscope was improved, an English scientist named Robert Hooke, and this is his photo here, Robert Hooke examined a very thin slice of cork made from a tree and looked at it under a microscope. So you can see in this picture, this is what a microscope looked back in the 1600s. When he observed it, it looked like an empty box-like structure which reminded him of a prison cell. And that's how the cell got its name, because he thought it looked like a prison cell. However, all cells don't look exactly like the square shape of a plant cell. Human, bacterial, fungal, and algal cells all take on different shapes. As those cells were studied over time, different observations and conclusions were made by numerous scientists. Together, their explanations became known as cell theory. It's one of the major theories in science. The major ideas of cell theory include three things, and you need to know and memorize these three steps. Number one, all organisms are made of one or more cells. Number two, cells are the basic units of structure and function in all organisms. And finally, number three, all cells come from cells that already exist. So this is the major proponents of cell theory. Today, cell theory is the basis that scientists use to study the parts of the cells and how they're organized, reproduce, and change over time. Okay, this is the end of our flip video for cells part 1. There are 2 links listed here. The first link is a Quizlet game that can help you identify the differences between prokaryotic cells and eukaryotic cells. It's got a couple of games, it's got some flashcards, it's a great resource to help you practice what you know. The second link here is to help the same kind of concept, except now it's for taxonomy, so the different kingdoms and domains and what those different things mean. So I'd encourage you to go to those 2 websites and practice playing the games, and make sure that you go back about 24 hours after the end of this video to do your House of Rigor questions. Remember, you needed 2 level 2s and 2 level 1s, and then also make sure that you go back and write your summary, 3 to 5 sentences. See you in class.